I'm going to invite the first speaker, and the rest will have to get us along the way. The other speakers are also joining soon. So I'm going to introduce her, and then I'll leave the floor to her, and then she can share what most of us have been waiting for all this long. Let me just get her biography, which is very interesting. So most of us know Pearl. We used to know her as Pearl. Right now, she's Dr. Pearl Magala, a postdoctoral fellow at the University of Washington School of Medicine, that's in Seattle. And her research is focused on understanding how bacteria infect human cells with a goal to develop novel therapeutics as antibiotics, as becoming less effective to the rise of multi-drug resistant bacteria. Some of these things you would have to break it down for some of us who are from another skill set. Paul received a PhD in chemistry from Johns Hopkins University, that's Baltimore, Maryland, and her BA is in chemistry from Mount Holyoke College, South Hadley, Massachusetts. So Paul, you're more than welcome. Let me hand over to you to share with us on the science of COVID.
Thank you, Jonathan, for organizing this, and thank you for the kind introduction. Can everyone hear me okay? All right, I'll take that as a yes.
I think I can.
You can, okay, good. I would like to share my screen. Jonathan, would you give me permission for that? Or make me like a co-host, something like that. All right, cool, thanks. All right, can everybody see my screen okay?
Yeah.
Okay, great, thanks. All right, so today I'm going to be talking about the coronavirus disease 19 and how vaccines work to provide protective immunity against the virus. So the first part of my talk is just going to be biology, and the second part will be the mechanisms of vaccine action. So where did we get where we are right now? So the coronavirus disease 19, also known as COVID-19, was first detected in patients who had flu-like symptoms and viral pneumonia in Wuhan, China, around December 2019. And the interesting thing is that this cluster of patients had all visited a wet market selling bats and pangolins. And sorry, my phone. I can't talk right now, sorry. Technical difficulties. All right, so yeah, this market had been selling bats and pangolins, among other things. And the reason I bring this up is that when the virus from the patients was sequenced, the virus is called SARS-CoV-2, which causes COVID-19. It was found to be very similar to the virus, the coronavirus in bats and pangolins, suggesting that these species were the source of the virus. Of course, now there is another theory that the coronavirus might have originated from a lab at the Wuhan Institute of Virology, but those investigations are ongoing. All right, so most of us probably first heard of a coronavirus probably just 1.5 to 2 years ago, after the outbreak, but coronavirus research has been going on for decades. And in the next few slides, what I would like to impress upon you is that the vaccines we have right now to fight the coronavirus want a week's worth of work, but rather decades of research. And I hope these next few slides will help you develop the confidence in the scientific community and in the vaccines that we have, because it's taken a very long time to get here. The research did not start only 2 years ago. So the first coronavirus was discovered in 1931 by June Almeida, who is a Scottish virologist, and David Tyrrell, who is a British virologist. They first isolated the coronavirus from chickens in 1931, and that's called the IBV virus, the infectious bronchitis virus. And contrary to what many of you had, the coronavirus is not named for the beer Corona, but rather it is named for this halo that extends off the sun's surface, which is known as a corona. That's kind of reminiscent of the spike protein that extends off the coronavirus. Could someone mute themselves? There's some background noise. All right. So in the next 30 years that followed, 2 more coronaviruses, these are called human coronaviruses, HCOVs, were discovered. So if you've had a cold during the winter or what have you, 15% of those colds are caused by human coronaviruses. So chances are, if you've had a cold, you've probably had a coronavirus infection. All right. So around the time that we were born in the mid-80s, there were so many advancements in coronavirus research, specifically the molecular biology. So at this time, scientists were learning how do these coronaviruses assemble their proteins, how do they replicate, things like that. And of note, the first genome of a coronavirus was sequenced. So the mid-2000s were kind of exciting on the molecular biology front of coronaviruses. So things like electron microscopy allowed us to image the virus for the first time. And in the early 2000s, there was the first outbreak of the SARS-CoV, again in China. This is the severe acute respiratory syndrome coronavirus. In the mid-2000s, 2 more coronaviruses were discovered, human coronaviruses, like I mentioned, that cause the common cold in about 15% of cases. And then around 2012, another coronavirus emerged. This is called the Middle Eastern Respiratory Syndrome coronavirus. And the source of this was thought to be camels. It was in the Middle East. Now, the interesting thing about the SARS-CoV and the MERS-CoV is that they're far more lethal than the SARS-CoV-2, which causes COVID-19. 15% of people who get SARS-CoV or MERS-CoV will die, in comparison to just 1% of people who get SARS-CoV-2, at least of last year. These percentages are changing with the emergence of new variants. So yeah, my point to take you through this timeline is to show you that, scientific discoveries and research have been happening on the coronaviruses long before the COVID-19 pandemic. All right. So to understand how to develop or treat COVID-19, you need to know the coronavirus structure. So most of us are used to seeing this ball here with the red spikes on it. If you take a cross section of it and look at it from the top, what you would see is a particle that looks like this. So inside the coronavirus is its genetic material tightly packed with other proteins, okay, that these form the core of the virus. And this core is surrounded by an outer membrane that has other envelope proteins, membrane proteins. And of particular note is this spike protein. It's really, really important in the coronavirus infection. All right, so that is what the coronavirus looks like. How does it cause an infection? So as I mentioned, the coronavirus has this thing called the spike protein, shown here in orange. The spike protein is recognized by another protein called the S2 receptor. This S2 receptor is found on the epithelial surface of your organs. So the S2 receptor in particular is richly expressed in your lungs, which is why when you get COVID-19, you have trouble breathing, and sometimes you can get respiratory failure because S2 is very common. on the lungs, but it's also found on other organs like the kidneys, your heart. And so if the coronavirus infection persists, this leads to multiple organ failure and then death. Okay, so once the coronavirus is recognized by this S2 receptor, the spike protein of the coronavirus is sort of like the key, and the S2 receptor is like the door. So it unlocks this door and gets inside your cells. And when it gets inside your cells, it releases its genetic material. This RNA replicates, and then also this genetic material encodes for several proteins that make up the coronavirus. So those proteins are translated. So once the virus is replicated inside your cell, and this makes like so many copies, the proteins are assembled and packaged, and the virus is released outside of your cell to go infect other cells. So I'm going to talk about the variants that you hear when you hear the Indian variant, the Brazilian variant. Those start to occur inside your cell when the virus is trying to replicate. The replication process is not perfect, and so it essentially makes these mistakes that we call mutations. And when mutated viruses start to leave your cell, then you can infect them with other people, and yeah, it's a whole different story, but this is where the mutagenesis of the virus arises. So what happens when the virus is released out of your cells? It encounters this thing called the immune response. This is your God-given army to protect you from viruses and bacterial infections. So once the virus is released outside of the cell, it encounters a soldier. One of the soldiers in your army is called the antigen presenting cell. This cell basically chews up the virus into tiny bits of particles, and then starts to once it chews up the virus, it presents those viral particles to another soldier in your army called the T helper cell, which activates other soldiers, the B cells and cytotoxic T cells. So the B cells are really cool. These are the guys that start to secrete antibodies against the virus. And the one thing I want to point out is that when you look at the virus here, it's engulfed by all of these antibodies. And guess who they are binding to? They are binding to the spike protein. Why are they binding to that spike protein? Because if they do that, then the virus cannot bind to the S2 receptor. It can no longer enter your cells and replicate. The cytotoxic T cells kill any cell that is infected with the virus. Now, something more that is really cool. Once your immune system cells, the B cells, T cells, encounter this virus, they do not forget who it is, okay? They will remember, so that the next time you're infected with the virus, they've already figured out how to fight it efficiently, okay? And so that is what we call immunity. Now, one of the questions people have is that why do some people get infected with the coronavirus and don't show symptoms, so asymptomatic people, or some people show mild symptoms, and then some people it kills them, right? So what's the difference? The difference is that asymptomatic people or people with mild infections are very efficient at clearing up the virus. If your immune system is weakened because you have cancer, HIV, diabetes, obesity, things like that, then you're not as effective at clearing up the virus, and it goes on to infect other cells and replicates, and yeah, and goes on and on. All right, so I'm going to switch gears here and talk about vaccines. A lot of people have questions about vaccines, how they work. It's kind of like this big mystery which creates fear, and so I want to demystify some of that. And I find these questions kind of baffling because all of us have been immunized at one point or another, and none of us were like, well, what's that polio vaccine doing, right? We just kind of went and got it. But I also understand, the fear, because COVID-19 is a new disease and the vaccines are brand new. But as I mentioned, the research has been going on for a very long time, and none of this was developed overnight. Okay, so the first vaccine that I want to discuss is the mRNA platform vaccine. The two vaccines that have been manufactured that use the mRNA platform are the Pfizer vaccine and the Moderna vaccine. These are distributed in the United States, parts of Europe, and other countries in North America. So how do these mRNA vaccines like Pfizer and Moderna work? So what scientists do is get a particle off of the vaccine. They get the spike protein. So they manufacture the gene, the mRNA that codes for the spike protein only, okay? And then they insert that mRNA, that genetic material, just for the spike protein, into a vesicle called a lipid nanoparticle, okay? So I kid you not, the lipid nanoparticle and the mRNA gene is the vaccine. That's it. When you go to get your shot, it's this yellow thing and this purple thing. That's all the vaccine is. One conspiracy theory I've heard about the vaccines is that they're like injecting particles that are supposed to track your movements and things like that. Yeah, what they're talking about is this lipid nanoparticle. But the reason you need the lipid nanoparticle is that mRNA cannot get into the cell by itself. It's negatively charged. You need something that is, I'm going to say, hydrophobic, that can interact with your cell to deliver the mRNA. That's why you need the lipid nanoparticle. All right, so when you go to get your vaccine, the lipid nanoparticle fuses with your human cell and then releases the mRNA. These are the instructions for making the spike protein into your cell. Your cell has all the machinery necessary to manufacture the spike protein. And when the spike protein is manufactured, it is released outside of the cell, where it encounters your God-given army, those immune cells that I spoke about. And this is a really cool example here, showing antibodies that are released, or rather manufactured, upon encountering the spike protein. So when you get your vaccine, you already have these antibodies around, ready to fight the virus should you get the infection. Okay, so I mean, basically the way the virus works is sort of the same way the vaccine works, except that the vaccine will not make you sick. So the mRNA has really been quite a miracle, actually, because these vaccines are so easy to scale up in terms of manufacturing. Okay, but the drawback is that mRNA is unstable, and so these vaccines need to be kept at very cold temperatures, so minus 70 Celsius. And so deploying these in developing countries is a bit of a challenge, which is why Pfizer and Moderna are not in Uganda yet. All right. So the other type of vaccine is the viral vector platform, and the vaccines that use the viral vector platform are the AstraZeneca, which is the vaccine being given in Uganda right now, and in the US we have a version of that from Johnson & Johnson. So how do these viral vector vaccines work? How does the AstraZeneca vaccine work? So the principles are the same as I just described for the mRNA. So again, the gene encoding for just the spike protein, remember that's the protein the virus needs to get into the cell, the gene for the spike protein is manufactured, and then that gene is put into another virus called an adenovirus, yeah. So adenoviruses cause common colds and things like that, but scientists essentially remove all the harmful genes from the adenovirus. And so whatever is left is harmless. So this adenovirus is harmless, and the only thing it's carrying is the gene to express the spike protein, the coronavirus spike protein. Again, the rationale here is to try and deliver the mRNA from the spike protein into your cells. And we know viruses are very good at getting into your cells. So hey, why not use them for something actually good? All right, so again, once the adenovirus is injected into, rather gets into your cells, and then does the same thing, starts manufacturing the spike protein, and the spike protein is released outside of the cells, and then elicits an immune response. So I hope you can appreciate the similarities of the principles between the two vaccines. They accomplish the same thing, just in different platforms. I think that's all I have prepared for today. Thank you for listening, and I'm happy to take questions.
Wow, thank you, Paul. At least now we know what spike protein is.
Yeah.
Does anyone have a question? Yeah, there are people who are raising hands.
Yes, I have a question for.
Ooh, did we lose her?
Okay, I have a question. I don't know if we're going to be picked or if.
Oh yeah, go for it. Go
Okay,
for
yes.
it. Yada.
Yes. Hi, Paul. How are you?
Good. How are you? Good to see you.
Good to see you too. I liked your presentation.
Thank you.
I don't know. I've come in a little late, so in case I'm asking something that has been addressed, please excuse me for that. But I wanted to ask. One of the concerns we are having here at home when it comes to vaccination is their ability to form clots. Okay, it's a side effect, yeah? Just like every medicine has a side effect. But could you talk a little bit about that in case you haven't? Because for some people, it is a real scare as to why they don't want to get vaccinated.
Yeah, that's
the fear of the clot, yeah, the fear of the clots. And also, what are the common side effects if someone got vaccinated, so that when they recognize them, they know this one is okay, I don't have to worry, but with this one, I need to seek urgent attention.
Okay, yeah. So I'm going to put a disclaimer here. I am not a physician. I'm just a PhD, so I'm more comfortable talking about the basic science. But okay, the one thing I can talk about is the clots. Like I showed, I think I'm going to go back to the AstraZeneca vaccine, since that's what's more relevant to Uganda. So the vaccine itself, which is, the vaccine itself, which is just this guy. I don't know if you can see my cursor yet. Yeah, I think you can. This in the strictest definition is the vaccine. But when vaccines are being manufactured, you need to add things like preservatives, adjuvants to really have a robust immune response. I truly believe that that's where the clotting problems come in. And that's, typically the case for any vaccine, not just the COVID-19 vaccine. There's really no good answer for that, but what I would like to impress upon people is, like, the AstraZeneca vaccine has been given to millions of people, millions, and the cases for getting clots are so small compared to, the people who actually died from COVID. The other argument I like to make is that COVID will give you clots also. And so I think this is the case where you sort of have to choose your poison, unfortunately. In terms of symptoms, like I said, I'm not
to be losing you.
Ooh. Is it okay now? Can everyone hear me okay now?
Yes, we can. Kindly proceed. We can hear you.
All right. Yeah, in terms of symptoms, I can't answer that. I'm not a physician, but I think we have a physician on the panel, so I guess he will be more well equipped to answer that question. Let's see, does anyone have any more questions? There were a few hands up.
There are some questions in the chat though. Maybe someone could read them.
Yeah, that'll be great, because I'm very bad.
Okay, let me go ahead and read some.
Okay.
Okay.
Yeah.
Let me just read what I'm seeing here. First of all, everyone is very appreciative of your presentation. So here we have a question. Does the release of the spike proteins in both vaccines keep going on in your body for a while?
Timeline. So the idea is, I'm sure the process does stop at some point, but the idea is that I think the key point from that is that the immune response generated by the spike proteins has memory. Okay, so those memory cells I'm telling you I talked about, where are they? If I can go through my slides. Yeah, these memory cells are what are providing you with the immunity. But is the spike protein continuously manufactured forever and forever? No, I don't think so.
Okay, then there's another question here. How exactly does the virus impact people with underlying issues?
Ooh, yeah, I did talk about that a little bit. So people who have cancer and other sorts of, I guess doctors call them comorbidities. It's because your immune response is too weak. And when your immune response is weak, the virus wins, right? Because then it gets to just keep replicating. That's what differentiates people who don't get sick from COVID-19 from people who die from COVID-19 because they have underlying issues like cancer or HIV. It's all about how efficiently you clear the virus.
Do you have any idea as to how the quenyokaza would work in a scientific way? Is there any way you can place the quenyokaza which they are pushing in Uganda in this science?
To the extent that wow, I'm really stretching here.
Yes.
Scientifically, I don't think I can explain it, but I think Ziada will have something to say about this later. Maybe sort of helping your lungs out, but yeah, I can't explain that from a basic science perspective.
Okay.
Any more questions in the chat? Yeah, now I can see the chat. Let's see. How exactly does the virus? Okay, that was Andrew's question. All right, I think we're through with questions, but I'm happy to take some more. I guess Jonathan's the timekeeper, so if we have time, I can take a couple more questions.
What I would suggest right now, we can move on to the next speaker, and then we'll definitely get some time towards the end.
Sounds good.
But Paul, thank you very much for educating us, and please stick around. We most definitely still have questions for you.
All right, thank you for having me, and thank you for organizing, Jonathan.
Most welcome.